we're going to look at how the first step of the light dependent reactions. The first step is water comes into the plant. Water comes into the plant through the stomata, which is underneath the cell, the little pores underneath the leaf, which is guarded by the which opens and closes with the use of a guard cell. Water comes in. Water is taken into the chloroplast, and it is hit by light. Light is absorbed by the pigment molecules and then kind of amplifies and goes in. Water, as soon as the water molecule comes in, the water molecule is broken up. This is the equation that it breaks up into, and 2 electrons are released. These electrons then pick up the energy from this photon. The light particle is a photon. Light energy is picked up by the photon, and that amplified, it picks up the energy, jumps up, and then there is a primary acceptor. The primary acceptor picks up this high energy electron. Remember, in cellular respiration, these high energy electrons were picked up by NADH. It's picked up by high energy electrons, goes through a cytochrome complex, similar to the one in the cellular respiration, where they have the cytochrome C complex in the membrane. Goes in. That is picked up by, and then it's absorbed by a molecule called P700. The first one is P680. So it starts with PS2 and then loses a little bit of energy. Some ATP is produced, some of the ATP is produced, and then it's picked up by P700. At the same time, the photosystem is also has a photon coming in. The chlorophyll molecules pick it up, it's amplified, the energy is amplified, the electron, whatever energy it lost here in the to make ATP is picked, then that electron picks up the energy from the photon, jumps up, is picked up by another primary acceptor, slightly reduces, and it is an enzyme called NADPH reductase. Reductase, meaning it is an NADP plus reductase. It is doing the reaction of reducing, which is reduction is reduction is gain. So just let's write this down here. Can't write it there for some reason. Reg. So reduction is gain of electrons. So it's, and it facilitates this reaction. So you have 2 E's that have come there, right? You have these 2 H's that are there. So that one combines. You have the H pluses that were there. Oh, sorry. Just for the second molecule. And then NADP plus together, it's going to make a molecule called NADPH. So now the high energy electrons have been carried through this, and this will be used in the light independent section to make the glucose molecule. Just to recap, light comes in, the water molecule is split. As the water molecule is split, this O2 is generated, it leaves the system, 2 electrons are generated and 2 H pluses are generated. The electron then absorbs the amplified energy from the photon. Remember when we were talking about the photoelectric effect? Jumps up, it kind of jumps to the next orbital, jumps up, it's accepted by a primary acceptor, goes down the cytochrome C complex. The cytochrome complex, as it's going down, it's actually losing some energy, and that energy is used to do substrate level phosphorylation and make some ATP. Goes in, is accepted by P700. At the second photosystem, photosystem 1, more light is coming in, there's more photons coming in, the energy is whatever energy it lost here, it picks up again here, is picked up by a primary acceptor, goes down a chain is acted on by NAD plus reductase. The word reductase means that it is adding electrons to something. So remember those things, oxidation is loss and reduction is gain. So it's adding an electron. Once it adds an electron, you are now producing NADPH. In cellular respiration, which was a catabolic process, you produced NADH. And in photosynthesis, which is an anabolic process, you're producing NADPH. The P will tell you if it's anabolic or catabolic. So there is a difference in the type of cellular respiration, photosynthesis you have. Sometimes there is something called as a cyclic. This is called cyclic. Cyclic photosynthesis. In a cyclic photosynthesis, it just means that it goes goes in the form of a cycle. In the cyclic photosynthesis, only photosystem 1 is used. It produces some ATP when required. So this process is essentially used to produce some ATP in case of a need for it. Now we're going to look at the light independent reactions. So when you say light independent, it means that it does not require light. This does not mean that it only has to happen in the dark or happen in the night. It can happen at any point of time. This just means that it is happening without the use of light. In the first stage, we needed light to create these high energy electrons. The whole point of the first part of the reaction, the light dependent part of the reaction, is to create these high energy electrons so that this molecule can be created. That molecule then is getting into the cycle and providing the required energy for the next cycle to take place. 3 CO2s in the light independent reaction is also called the Calvin-Benson cycle. The first step is for 3 carbon dioxide molecules to come in. 3 carbon dioxide molecules come in, and then there is an enzyme called Ribisco. Ribisco is an enzyme. Ribiscus enzyme, it is one of the most abundant enzymes in the world. If you look outside, you see all the trees and all the leaves. Every leaf has a bunch, is green in color, which means it has a bunch of chlorophyll molecules, and it does this reaction. So all of them tend to have the enzyme Ribisco. Just like in cellular respiration, where you had oxaloacetate, which was a molecule which was recreated. In this, you have RUBP, which is a molecule that is regenerated. So these two connect, and it goes through multiple phases. I'm not going to expect you to remember these things. These are not required. And is able to make 2 G3Ps. We had a similar product created in cellular respiration where you had the phosphates, where you had the glucose break up into 2 G3Ps and then go through multiple processes. This is literally the opposite of that reaction. In that reaction, you had acetyl-CoA come in and join oxaloacetate. In this reaction, you have 3 CO2s coming in and joining RUBP. In that reaction, you had oxaloacetate being regenerated. In this reaction, you have RUBP being regenerated. The end of this reaction, the halfway point of this reaction, you're going to create 2 G3Ps, which is glyceraldehyde 3-phosphate. And then these 2 G3P molecules combine together, the phosphates come off, and it produces 1 C6H12O6. In cellular respiration, 1 C6H12O6 breaks up. The first breakup that happens, it creates 2 G3Ps. So these processes are actually going in the opposite direction of one another. this is a very good image for you guys to kind of remember, so you can answer questions on the test, and also like to understand how photosynthesis takes place. So the first part of photosynthesis is light reactions, where it comes into photosystem 2, goes through the electron transport chain, which is a cytochrome C system, then goes through the photosystem 1 and goes through a short electron transport chain, where then it meets with NADP plus reductase, becomes NADH, and some ATP is produced in this process. The ATP and NAD plus NADPH goes into the light independent reactions, where the CO2 comes in and combines with RUBP at the halfway point of that reaction, that is before the regeneration of RUBP. 2 G3Ps are created, and in the process NADPH is oxidized, oxidation is lost, is oxidized into NADP plus, and then it goes back into the light dependent reactions. ATP is used up and ADP goes back into the light reactions. Some of the glucose that is produced is then combined with a fructose molecule, which is just an isomer of glucose. It's combined with that to make sucrose, and then that sucrose becomes a part of fruits or whatever the plant is putting out in order to export its seeds to other places. And then some of it is stored in the form of starch. Starch, as you learned in chapter 5, is nothing more than a long chain of glucose molecules where they do a dehydration reaction and a polymer is formed. So this is just a basic overview of photosynthesis. You guys, you'll have very few questions on photosynthesis in this on this test, or any test for that matter in this class, because you will do photosynthesis more in detail in your next biology class. The main reason I took about 35, 40 minutes to do photosynthesis is so that you understand the difference between the two processes. Cellular respiration is incomplete if you don't understand photosynthesis, and photosynthesis is incomplete if you don't understand cellular respiration, because these are processes that feed into each other. Cellular respiration feeds into, photosynthesis feeds into cellular respiration by producing C6H12O6 and some oxygen, and cellular respiration feeds into photosynthesis by producing CO2 and H2O. So round and round the cycle goes. So I'm going to change the order a little bit. We're going to do DNA replication and then go back and do mitosis and meiosis, so that you have a better understanding of how the DNA first replicates, then breaks up, and then the cell itself breaks up. So mitosis has 2 parts to it. There's the M phase, in which the DNA is separating. Before the M phase happens, that your DNA has to replicate. So if you have 2 strands of DNA, these 2 strands of DNA in the S phase, I'm just going to change the color of the pen. In the S phase, this strand has to make a copy of itself, and then this strand has to make a copy of itself. This happens in the S phase, and after the S phase, it goes into there's one more phase, and then it goes into the M phase. M phase is the mitotic phase, in which these guys are separated. So if this was your cell, let's assume this was your cell, these 2, I'm just going to put a joint here so you can understand that. These 2 will migrate to this corner, and these 2 will migrate to this corner. And then cytokinesis happens, where the cell will basically kind of split in the middle right there. And now one cell becomes the second cell. And that process is called mitosis. So before I get to mitosis, there are different schools of thought on how to do this. You can go from mitosis to replication. I just prefer to go from replication to mitosis. Whatever it is, we'll cover all of them. Just remember these two names. The DNA was discovered. Obviously, we had DNA for the longest period of time, and the DNA was discovered by Watson, James Watson, and Francis Crick. Dr. Watson, till recently, still taught at the UC system. So I had the privilege of meeting him. So DNA is the basis for inheritance. The characteristics that that everything in your body is governed by genes, and those genes code for different types of proteins. The color of your eye is because of certain genes you possess, and these things, when these can be transferred from one generation to another, and it is encoded in the chemical language of the DNA. The chemical language of the DNA is the 4 base pairs, A, T, C, G. Now, depending on how it is arranged, depending on where it is, depending on how it splits, all of that tells you what information is carried in your body. So I'm gonna go through a basic experiment. So there are many, before we discovered the DNA and we understood DNA, there was a huge debate on what is it that was the molecule of inheritance. What is it that makes a child look like their parents? All this so there are different schools of thought in that. There at one point of time, people thought it was proteins. I mean, way before that, there was the homunculi theory. There were ovohomunculi, there were sperm homunculi. There was like different types of theories that were prevalent at that point of time. One of the first experiments, and you can definitely expect questions on this on your test. One of the first experiments was conducted by Griffin. So what he did was he took 2 different types of bacteria, 2 different types of bacteria within the same group. The group was called a Streptococcus pneumoniae. Pneumoniae is what it is. Pneumonia causes pneumonia. Strepto, meaning it kind of is arranged like a grape cluster. And coccus, meaning round. So Streptococcus pneumoniae, there was 2 strains of this bacteria. One is the smooth strain and the other one is the rough strain. The smooth strain basically has a capsule. So it has the cell membrane. In eukaryotes, it is made out of phospholipid bilayer. In bacteria, it's a peptidoglycan layer. So it has the peptidoglycan membrane. On top of that, it has a smooth capsule that covers it. It's kind of a slimy capsule, and that provides it virulence. What do we mean by the word virulence? Virulence means it provides it certain characteristics that make it pathogenic. The word pathogenic, there are 99.99% of most of the bacteria that's in the world are non-pathogenic. They do not cause human disease. When something causes a human disease, it is pathogenic. So anything that confers virulence to a bacteria makes them pathogenic to us. So the fact that the smooth core strain is pathogenic because it is given virulence by the existence of the capsule, and then the rough, the one that lacks the capsule, is non-pathogenic. So he took a bunch of live, a very simple experiment. He took a bunch of live cells, S cells, injected into a mouse, and the mouse died. And then took a bunch of R cells and then injected into the mouse, and the mouse lives. So these 2 things are his control. For any experiment, you're going to need a control and a variable group, and then an experimental group. Sorry, and so he had his controls. And the next, what he did was he heat killed the S bacteria. When you heat kill something, proteins denature. Proteins tend to denature, so the bacteria is killed. The capsule is also made out of protein. Everything is denatured. And then he injected the mouse with heat-killed bacteria. Once he injected it with heat-killed bacteria, the mouse was alive. It was alive. Then what he did was he took a bunch of the heat-killed S bacteria, mixed it with R bacteria. Bacteria are capable of doing a process called transformation. That means if there is DNA, and only DNA, if there is DNA in its surroundings, it can pick it up and start using it. This is how sometimes you can have like a bacterial infection, and if you don't take your antibiotics for the whole whole duration of it, you can fall more sick, because the ones that are that have an immunity to the antibacterial, that are resistant to the, not immunity, that are resistant to those things, then can pick up DNA from their surroundings and become more resistant. The bacteria can also do plasmid, where they can transfer a part of their DNA to another bacteria. So because of the fact that they're able to do transformation, because they're doing transformation, they then are able to pick up the information from the DNA of the heat-killed bacteria, the DNA, the strands of DNA that codes for a capsule, develop a capsule around themselves, and then become pathogenic. So basically, it's that if this is your DNA, this is just floating in the environment, and this is the DNA that codes for the capsule. You have your rough bacteria here. It kind of takes this information in, forms what's called a plasmid, a separate set of DNA on the other side, and now it's able to produce And when this one is decoded and then it produces a capsule around itself, now the R bacteria has transformed into the S bacteria, hence killing the mouse. And once, because it's killed the mouse, we know, and then when you look at it, when he took a sample out of it and like grew it out, he found that the bacteria has a capsule around it, because that's essentially what happened. The process is called transformation. So he was able to ascertain that the change in genotype, there are two things. I don't know if you remember your high school biology. There are two things. There's genotype, which is the DNA, the gene itself. The genotype then codes for proteins and then makes a phenotype, is able to make a phenotype. Oh, that's the word. The phenotype. Phenotype is the expression of the genotype. So the R bacteria took in a piece of DNA from the heat-killed dead bacteria, makes a plasmid, and then when that is expressed, the phenotype is the production of a capsule. And then when that R bacteria is dividing and making more copies of itself, since it has now the DNA material to make the capsule, the next generation now all have capsules. So that's basically what he said, is due to the assimilation of an external DNA by the cell. And that process, this process is transformation. Horrible handwriting. The next set of discoveries that happened for us to understand that DNA is the key for heredity, is the molecule that carries heredity, is using bacteriophages. Bacteriophages are viruses that basically infect bacteria. There's a lot of modifications that have happened to our DNA in order to stop bacteriophages from cutting up the DNA. The viruses can attack the DNA and then cut up the DNA of the, attack the bacteria, cut up the DNA of the bacteria, put itself in the middle and make copies of itself. So viruses, this is a very standard image of a virus that you've seen. Usually they have an icosahedral, which is like a 20-sided. One class of them, not all of them have it, but you know. They have an icosahedral head, and they have this protein kind of a body, and they have their DNA on the inside. That DNA, this is then once it has receptors right there and its tails, and they kind of act again like lock and key mechanism. They kind of lock onto certain receptors on the cell. Once it does that, it basically takes its DNA and pumps the DNA into the cell that it is infecting. And then that DNA goes in there, finds its own, finds the DNA of the bacteria. Remember, unlike our cells that have a nucleus around it, the bacterial cell does not have a nucleus around it. It's just a nucleoid region where the DNA is found. So then this DNA migrates to wherever the bacterial DNA is. Let's assume that its DNA is right here. It's then going to kind of make an cut there, and insert itself into this region, and then make more copies of itself. So every time this part is expressed, because this is the viral DNA, once that part is expressed, the proteins required to make the phage head, this portion of it, to make the tail fibers, all that information is right here. It assembles a copy of itself. The cell breaks open, and a whole bunch of these viruses are then pumped into the atmosphere. The next very famous experiment is the Hershey-Chase experiment. In this case, what they wanted to find out was, is it proteins that infect the cell, or is it bacteria? Is it DNA? What is it that infects the cell, and what is it that is carried is the genetic material? What are the genetic material of the T2 phase, right? T2 phages are, again, they're phages, they're bacterial phages. They infect bacterial cells. What they basically did was, you can read this also, and I'm going to kind of break it down and explain it to you, like what actually happened. If you think about the protein molecule has one unique element in it, which is sulfur. Sulfur is able to make the disulfide bridges. So you have beta sheets, alpha helixes, and they make these, the secondary structure of protein, beta sheets and alpha helixes, and they make these disulfide bridges to hold them together. They're covalently bonded. Sulfur by itself, regular sulfur has, its atomic mass is 32. They used radioactive sulfur. When we say radioactive sulfur, it just means that it has more neutrons. This one has 35. That means it has 3 more neutrons than the one that you see on your periodic table. They took the phages, and they grew them in a medium that had radioactive sulfur in it. So in your lab, if you guys have done plates, the petri dishes where you grow the plate, you put the growth medium in it and grow bacteria in there. Whatever you put in that growth medium is food for the next generation. And so in your lab experiment, you would have taken a plate, done a smear right there, dragged that down there, done a smear right there, and then taken that and dragged it right there, so that here you get a single colony, right? So if you have like a single bacteria, then you get the clones of that in that colony. This colony will be pretty much clones of the one bacteria that was there. This medium that you use has all the materials required for the bacteria to grow. You might have used a tryptic soy agar, which is the most basic thing. Basically, they took the soy agar and then they put sulfur in it. so that there is sulfur available. That sulfur, the next generations, the generations of phages that were growing there, were using it as a part of its protein. They took the radioactive protein phages with the radioactive protein phages, and then they infected a bunch of bacterial cells, let them grow, and then basically centrifuged it to find out where that radioactive sulfur will be found. So when you centrifuge something, the DNA, we talked about this in chapter 6, when you centrifuge it, the DNA, which is a heavier molecule, goes in the pellet, which is in the bottom. So if this was your centrifuge tube, this is what the tube would look like. The DNA would be here in the form of a hard pellet, and then this would all be the liquid that contains all the other organelles, all the proteins and everything. So it kind of settles in along the course of density. So when they used sulfur, radioactive sulfur, they found it to only be in the liquid on the supernatant on the top. So that it was found only in the liquid. It was not found in the pellet. And then when they did the same experiment with the radioactive phosphorus. When they did that with radioactive phosphorus, again, there is no phosphorus in your sulfur molecule, in your protein molecule. There's no sulfur in your DNA. So by using sulfur and phosphorus, you clearly can distinguish between proteins and DNA. When they did the exact same experiment, they found it in the pellet, the radioactive sulfur in the pellet. So with that, they were able to conclude that the T2 phage DNA and not protein functions as its genetic material. And you can have questions on the test that say, why is it that we used sulfur? Proteins have carbon. Why not use carbon? Why not use nitrogen? One of the reasons we don't use carbon and we don't use nitrogen, we don't use oxygen and hydrogen and all of these, is because they are common in both these molecules. They're common for proteins as well as DNA. So if you used anything that was common, there would be no way of distinguishing it. Sulfur is something that's unique to protein here, and phosphorus is unique to DNA. By creating radioactive molecules that were unique to each other, you're easily able to track it. Let's look at the basic structure of a DNA molecule. Let's look at the basic structure of a DNA molecule. There is going to be, so the monomer of this is going to have 3 components. The monomer here is that, pen. That would be the monomer, right? That's the monomer. It's a nucleotide. There are 3 parts to it. One, there is a 5-carbon sugar called ribose. So we're going to talk about this in 1 second. The sugar itself is ribose. That's 5 carbons. So the formula would be C5H10O5. Five-carbon sugar. You have a nitrogenous base. And then you have a phosphate. All 3 of these put together is going to be called a nucleotide. This 5-carbon sugar in position 1, the first carbon is where the nitrogenous base attaches. In position 5, the 5th carbon is, so 1, 2, 3, 4, and 5. Here there is a carbon, and this carbon is where the phosphate is attached. This is the 5th carbon. The 5th carbon is where the phosphate is attached. The 3rd carbon, which is this carbon here, which is the 3rd carbon, is the one where the phosphate of one nucleotide, the phosphate that is attached to the 5th carbon of one nucleotide, is going to attach into the oxygen. So a dehydration reaction is going to take place. There's an OH. If this wasn't attached here, there's going to be an OH here. This OH is going to come off. This OH is going to come off, and there's a dehydration reaction that takes place, and it attaches here. The dehydration reaction will take place there, and then it forms an attachment in that location. The second carbon this is a unique carbon. For DNA is deoxyribonucleic acid. It is in this second carbon where one oxygen is missing. So this is what the 5 carbon would look like. Okay. Here you would, if it is ribose, which is RNA, if it's ribonucleic acid for RNA, this in the second carbon position, CH2OH. In the second carbon position, you're going to have OH if it was RNA. Because it's DNA, this O is missing. Hence, it is called deoxyribonucleic acid. Just to recap, RNA is ribonucleic acid. There are other differences, and we'll talk about that. One of the difference between DNA and RNA is in the ribose, in the second carbon position, instead of having an OH, it has just an H. And so the third of this, so that's the nucleotide. Just to recap the nucleotide, in position 1, you're going to have a nitrogenous base. In position 5, you're going to have a phosphate. Position 3 is where the attachment takes place. The dehydration takes place between 2 monomers and attachment takes place. So the 5 of one nucleotide, the phosphate of the 5 of one nucleotide is going to attach to the OH, the OH that is sticking out of the third one. It's going to attach there. So it's going to be a 3-5-3-5-3-5 or 5-3-5-3-5-3, which DNA strands are anti-parallel. So one goes 5-3, the other one goes 3-5. Between the nitrogenous bases, you're going to have hydrogen bonds. T is going to form 2 hydrogen bonds with A. This is the base pairing rule. Scientist by the name came up with this, Graf came up with this, and A is always going to attach to T with 2 hydrogen bonds. C will attach to G with 3 hydrogen bonds. These bonds are hydrogen bonds. If you remember from our previous chapters, we talked about hydrogen bonds. Hydrogen bonds are a weak attraction between something that's partially positive and something that's partially negative. DNA strands are anti-parallel. So one strand, you're going to have this side, the 5th carbon, 5 prime. The 5th carbon is unattached to anything. And on this side, the 3rd strand is unattached to anything. And the strand complementary to that is 3 is this way and 5 is this way. So think of them as, a good way to think of it is like a freeway. One side goes north, the other side, both of them look like they're running parallel, but actually traffic on them, one is going north and one is going south, so they're like anti-parallel. The term is anti-parallel. And then if you have an A on this strand, you'll have a T on this strand. If you have a C there, you'll have a G there. And this would be connected with 3 hydrogen bonds, and this would be connected with 2 hydrogen bonds. I'm not going to expect you to be able to draw these out or memorize them, but you definitely do have to kind of be able to recognize which one is a purine and which one is a pyrimidine. you have to recognize which one is a purine, which one is a pyrimidine. The two fused rings here. Why is this not drawing? The two fused are purines. I keep confusing these all the time between purines and pyrimidines. The way I remember is the shorter the name, the bigger the looking molecule. These are pyrimidine. These are actually bigger molecules, but they, when you draw them out, they kind of look smaller. So we talked about ribonucleic acid in DNA. A will always combine with T, G will always combine with C. So if I gave you a question that said 30% of somebody's DNA has T, that means the other 30% has to be A, and 20% has to be C, and 20% has to be G. The reason you know that is because if there is 30% T's, then it has to be 30% T's. And this is just an example. This is not anybody's DNA. When we talked about the sugar molecule, we talked about how one of the difference between DNA and RNA is that DNA has in the second position, it's going to have an H. In the second carbon position, it's going to have an H, whereas RNA is going to have an OH. The other difference is that thymine is not found in RNA. Instead, you find uracil. And uracil is the thymine molecule without this methyl group. So that's two differences. The third difference between DNA and RNA is that DNA is actually double-stranded, whereas RNA is single-stranded. DNA forms a double helix, so it's going to go that way, one strand, and that way the second strand. So if this strand is 5' to 3', this strand is going to be 3' to 5'. I hope I followed the line correctly. 3'. So they run anti-parallel. No, oops. They run anti-parallel too. So, oops. Nesselson and Stahl, basically they proved when DNA replicates, the way it replicates is a semi-conservative model. That means, what does this mean when we say it's a semi-conservative model? If I have two strands of DNA, which is what we have, two strands of DNA, first step is going to be to unwind. What do we mean by unwinding? When we say unwinding, first thing is to break those hydrogen bonds. There are hydrogen bonds. There are two hydrogen bonds holding A and T together. There are three hydrogen bonds holding C and G together. The first thing is to break those hydrogen bonds. Breaking those hydrogen bonds, they're not actual like covalent bonds and stuff like that, so it can be easily broken. So the first thing is to break that, and then I'm going to change the color of the pen so you guys can see clearly. This strand is going to replicate. When we say replicate, what does it mean? If there is an A here, a polymerase is going to come in and put a T there. If there was a C there, it's going to put a G there. So a new strand is created, right? And that's a new strand. And then that's going to replicate, make a new strand. And then this is going to replicate here and make a new strand. And then when it breaks, now you have two sets of, let me change the color again. So now you have 2 new sets of DNA. This is one set of DNA, and oops, and this is a second set of DNA. So for every time DNA is replicating, it is semi-conservative. What do we mean by semi-conservative? There's an old strand, and then there's a new strand, and there's an old strand and the new strand. So the way they've proved this was, again, by using radioactive nitrogen. Nitrogen basically has 14, its atomic mass is 14. Instead, that means it has 7 neutrons. In this case, they use something that has 8 neutrons by using an isotope of nitrogen, and then just basically following that nitrogen isotope, because it is in the nitrogenous basis. And by following those isotopes, because it is radioactive, you can easily see it and just following it. So before that, they had a conservative model. The idea of a conservative model would be that one strand is old and one strand is completely new. They also thought about a disruptive model. That is basically everything breaks up. There is some new parts to it and there's some old parts to it. But now we know that with the first replication and the second replication, I mean, you have to use different amounts of replication to figure this out. Just can't. They figure that it is semi-conservative. So when you are trying to replicate the DNA or copy the DNA, there has to be a remarkable amount of speed and accuracy, because if there isn't accurate, first of all, it has to happen fast enough so that the cells can make more copies of itself, and you cannot have your DNA just decommissioned for such a long time. The second thing is that it has to be accurate. If it is not accurate, it can lead to multiple number of mutations, multiple issues, and if your DNA replicating is not accurate, things in your body would change constantly. It also requires a large number of enzymes to take part in it. There are various types of proteins that are involved in it. And all of this in a eukaryotic cell actually takes place inside the nucleus. So these proteins have to be transported into the nucleus for these things to happen. The next step is a replication itself. It is a little more complicated, so I will do it in class. Please pay very close attention to the process and ask as many questions as possible so you kind of have a good understanding of what it is.